And so that is it for our very short senses. So we can get through all of those nice and quickly. And now, as promised at the beginning, I'm going to end us off in our individual systems with the visual system. So we started with a big one, we got through all of the smaller ones, and now we're going to end with another bigger system. So our visual system is all about our sense of sight. And our stimulus here is going to be light, specifically a form of electromagnetic radiation that travels as a wave. So electromagnetic radiation is the fancy way of referring to the specific spectrum that we are able to perceive with our eyes. Our electromagnetic spectrum actually covers quite a few other things. If I flip ahead here, this teeny little sliver here is our visible light spectrum. So that's the electromagnetic waves that are within the range that we can see. But those same waves, if they are sort of higher frequency, we can have things like X-rays or ultraviolet rays. If they are much lower frequency, they could be AM or FM radio, they could be radar or infrared rays. Those are all the same electromagnetic spectrum, it just depends on the frequency. And I always make a point here, if you have taken physics, you might be scratching your head about talking about light just as an electromagnetic wave, because light is a weird thing and it can be both a particle and a wave, but when it interacts with our eyes, it's behaving as a wave, so we're talking about it as a wave. And if you have not taken physics and were not even remotely thinking that, then just ignore all of that, and we're going to talk about this electromagnetic wave that is light. So a lot of the stuff that we've already learned about waves from talking about sound waves, they're still going to apply when we're talking about light in terms of waves. So we can still talk about amplitude. And so instead of with sound, amplitude was how much energy is in the wave, and therefore how loud the sound is. Here, amplitude is still how much energy is in the wave, but now it's related to brightness. So a high amplitude wave is very energetic and is very bright. We can also look at wavelength or frequency, and that's going to affect our perception of color. So if it is very high frequency, or low wavelength, we would have something that's blue. If it is very low frequency or high wavelength, it's going to be red. And so we can see anywhere from 300 and some odd nanometers for our wavelength up to 700 and something nanometers. I don't want the specific numbers, just knowing that we're in the range of nanometers is perfectly acceptable. And we can even talk about purity, kind of like when we were talking about the quality of sound. In light, we can also have purity, which is going to give us an idea of the saturation or the richness of our colors. So at the top here, we have 100% saturation or very pure light, down to 0% saturation or very impure light. And so if we're going to move on from there, we can actually start talking about the structures within the eye. So this is our large-scale map here. And the same as with the ear and all of the other stuff, I'm not going to show you an image and ask you to label it, but you would be expected to know which order of structures does a ray of light pass through as it enters the eye. And so we can look here and we can walk through sort of step by step what is light passing through. So if we were looking at this butterfly, as we're coming in, we have this outer layer, this sort of clear coating on our eye, and that's our cornea. So if you've ever had, say, a scratched cornea, maybe you got dust in your eye and scratched your eye and caused some damage, it's that outer layer of your eye, the cornea, that's damaged first. What you would keep going, and the gaping hole, the black spot in the middle of your eye, is your pupil. And the pupil, the size of that pupil, is determined based on the iris, the colored part of your eye, and the ciliary muscles that control it. So these muscles can cause the colored part of your eye to stretch or expand. And so if the iris is pulled back, then your pupil gets larger. If it's relaxed out all the way, then your pupil gets smaller. So this combination of your iris and these muscles controlling it determine how much light you let into your eye. This is why when you wake up in the morning, if you turn the lights on too quickly, everything seems really bright, and then your eyes, you have the big dark pupils. That's because your pupil was wide to let in lots of light in the dark. And then once you're out in the sun, the pupil gets smaller because you don't need as much light because there's tons of light outside. So that's going to adjust and change how much light comes into the eye. So after we pass through the pupil, we're going to enter the lens. And the lens is just like you would think of a lens being. So the lens of your glasses or the lens of a magnifying glass, it's going to be something that focuses and manipulates the light that's coming in. And so in this case, our lens allows us to focus the image or the light coming in on the back of our eye. And this surface, this sort of screen on the back of our eye, is called the retina. And the retina is where we're going to be picking up on all of the information that's coming in the form of light. And interestingly, you may notice that the image, when it hits the retina, is actually inverted. So our eyes actually perceive the world upside down. And our brains just sort of correct for that internally. And there's some very interesting research that's gone around where if you wear glasses that invert the image before it gets to your lens so that it ends up at the back of your eye right side up, which is upside down to normal, your brain takes about 3 or 4 days and then just figures out what's going on and nothing seems weird, even though the world is coming to your brain upside down. We're very adaptable that way. And that's a cool thing to look up if you want to waste some time. At the very back here, almost directly behind sort of our pupil through the lens to the back of the eye, this is our fovea. That's our focal point. That is where we have the most accurate vision. And then our next point of interest is going to be this strand here, and this is our optic nerve. This is where all of our receptors are going to send their signal out to the brain. And interestingly, even though our optic nerve is exiting the back of the eye very, very close to our fovea, which is our most accurate point of view, it still interferes with our vision. So we have a blind spot, a place where we can't have any receptors, because that's where all of these nerves are exiting the eye. And our brain does a very good job of glossing over the fact that a blind spot exists, but we do have a very prominent blind spot fairly close to the center of our vision. So again, if you take a look at the textbook, there's an image that shows you your blind spot by getting you to focus on 2 dots on a page and adjusting where the textbook is positioned until one of those spots disappears. It's very, very cool to try, just very difficult to do on a screen. So take a look at the textbook or maybe Google that image and then lift your laptop around and see if you can make it work, because it is very cool. So moving on from there, the textbook stresses that the eye serves 2 main purposes. It channels light to the neural tissue that receives it, so it gets light to our retina, and they house all of the tissues that are involved in doing that receiving. And I find it strange that they specify that those are the two purposes. So the eyes get light to the place that you can see, and they house the thing that lets you see. For more useful information, we can go over in words what I just did on the image. So if we talk about what's happening, our light is entering the eye. It's going to pass through the cornea, that outer coating on the eye, and then through the lens. So it has to pass through our pupil to get to the lens. And our lens is capable of adjustments. So these adjustments are called accommodations. And all it means is that your lens can get sort of shorter and fatter or longer and skinnier based on what you're trying to focus on. So if you're trying to focus on something that is close to you, say you're looking at your computer screen right in front of your face, then the lens will get fatter and rounder to give you a clear image of that thing that's up close. When you're focusing on something further away, maybe you're going to get up and go for a walk, and you look across the room, you're looking at a distant object, and the lens will flatten out, so it will get longer and skinnier to let you focus on those further away things. And so for those of us who have difficulties seeing either up close or far away, it's usually because our lens has trouble with these accommodations. So you can have trouble focusing on things that are up close because your lens won't get rounder, or you can have trouble focusing on things that are far away because your lens won't get narrower, or you can have both if you're very unlucky. And so we've already talked about the fact that the eye can make adjustments to alter the amount of light that reaches the retina. So this is your iris, that colored ring of muscle around the pupil that's going to interact with that ciliary muscle to regulate the size of the pupil. And the pupil is the black, empty space in the center of the eye. And so this all works together to regulate how much light enters your eye, how much light you need to see. Now we can take just a brief moment to talk about something strange that our eyes do, and these are called saccades, which are very small, constant movements of the eye. So if you've ever tried to focus on someone's eyes while you're having a conversation with them, you might notice that their eyes dart back and forth every once in a while. And that is, you're observing their saccades. And so these movements, these little adjustments, are very critical for good vision, but they can be somewhat unhelpful because they can sort of glance quickly towards something that you're interested in but not necessarily trying to look at, so they can give away what you're paying attention to. And it's something that we can't really control. These movements are going to happen kind of all the time, no matter what. All right, so next we can talk about the retina, and so these are going to be the neural tissues lining the inner back surface of the eye. So we're calling it a neural tissue because it's made up of a bunch of receptors that are going to feed into the neurons that are sending sensory information to our brain. So the retina is involved in absorbing light, and it's going to start processing the images that we're seeing, mostly telling us sort of the intensity or the colors that we're seeing, and sending that information to our brain. And so if we look at the image that we had before, this is our retina. This is what it looks like if we zoomed in on the retina. And so we can see here that the light is still coming this way, sort of from the left side of the screen to the right side of the screen. But to get to our sensory receptors, to get to our rods and cones, this photoreceptor layer, the light has to pass through all of these support layers. And if we were taking a anatomy course or a sensory specific course, we could go into all of these cells, the ganglion cells and the bipolar cells, and all of these components that help us see and process what we're seeing. We'd go into that detail, but for now, we're just aware that there are other cells that are supporting and working with our photoreceptor cells. But we're going to care mostly about these receptor cells. And like we've said now, there are two different types. We have cones and we have rods. And the cones have this sort of conical shape and the rods have this sort of rod-like shape. And they are located at the backside of that retina layer. So it's always interesting that light has to pass through all of this other junk to get to our receptors. All right, so for our two types, our cones are specialized visual receptors that are important for daylight vision and color vision. So these cones, I always remember which ones are responsible for color because cones and color both start with C. And cones need a lot of light to function. So they really only work in daylight when we have tons of light available. And this is why when you're looking at stuff at night, it's hard to tell what color things are because our cones only really work well during the day, and they're how we get our color vision. So our cones tend to be concentrated most heavily in the very center of our retina towards that fovea, and they're going to fall off in density as they get further away from the fovea. And we have a graph coming up that'll show us how these are oriented. And so the fovea is that small center point, that small focal point that only has cones. There are no rods in the fovea. And that fovea gives us our greatest visual acuity. That's where we are most sensitive and most accurate at seeing things. And I'm actually going to skip this slide briefly to go to this image, and then we'll come back and talk about rods. But if we look at this graph here, we're going to focus on this red line because the red line is showing us our cones. And so if we look at what it's telling us here, so we have angle from the fovea. So we're basically looking at the eye. If this is the front of the eye and that's our lens and that's our cornea and light's coming in this way, our fovea is right at the back of the eye. And as we move further away, we're getting higher in degrees away from that zero start point, and then it counts up on the other side as well. So this is looking at the very back of our eye, and then wrapping around towards the front, and then wrapping around the other way towards the front. And then this axis is just how many receptors. So higher means more receptors. And so we see that our cones are concentrated very, very strongly in the fovea. There's only cones in our fovea. And then they drop off. Our concentration of cones goes down really, really low. And once we're away from the fovea, there are very few cones on these other parts of the eye. There's very few of them over here. There's very few of them over here. But there's tons in the middle here, right around that fovea. So we're really good at telling what color things are when it's close to what we're looking at. And this is something that our brain kind of tricks us into not realizing. But if you pick up a pen, say you have a red pen on your desk, if you hold that over so that you can only see it out of the corner of your eye, it might become very difficult to tell that that pen is still red. You can still see the pen, but the color becomes harder to detect. And that's because we don't have many cones in our periphery. But if we go back to talk about our rods, our other kind of visual receptors, these ones are specialized to play a role in our night vision, and they're really good for our peripheral vision. So our rods do everything that isn't color. They don't need lots of light the way that cones do, so when we're looking at things in the dark, it's our rods doing most of the seeing. And in this case, the rod density is going to be the greatest just outside the fovea, because the fovea only has cones. And again, they're going to gradually decrease as they get away from that fovea, which we can see here. So at the fovea, our blue line now is our rods. There are none at the fovea. And then there's tons just outside of the fovea, and then they slowly decrease outside of there. But you'll notice that there is far more area under the blue lines than under the red lines. So we have far more rods than we do cones. They're just not located right in the middle of our vision. They're not located in the fovea. They're located everywhere else. And so if we're talking about the fact that our cones do color and daylight vision, and our rods do everything else and night vision, there has to be a point where we switch from using our cones to using our rods. And so the process where our eyes switch over to dimmer light is called dark adaptation, because we're adapting to the dark. And so the idea here is on this axis, we're going to be time in the dark. So at 0, that would be you were in a bright room and someone flipped off the lights. And this axis is sort of a vague thing saying intensity of light to produce vision. And that's how bright does something need to be for us to be able to see it. And so right at the beginning, at this 0 time, right when the lights have been flipped off, we're still using our cones because that's what we would have been using while the lights were on. And things would have to be very bright for us to see them. So when you first turn the lights off to go to bed at night, maybe it's really hard to see where you're going because it's very, very dark. So you stumble around in the dark a little bit because you can't really see anything. And that's because nothing is bright enough for your cones to pick up on. We have this very quick changeover in the beginning where our cones are starting to sort of turn off because they're not very helpful. And right as we get to this 10-minute mark, we switch over to our rods. And our rods need much less light than the cones did. But it's a gradual switchover. So we have this big decline by the time we get to 10 minutes, where we've switched over to using our rods, and we need far less light at this 10-minute mark than we did up here at the 0 mark. But we're still not fantastic in the dark. So that's, the first 10 minutes after you flip the lights out to go to bed, it's still very dark to you, but it's not as bad as it was right at the beginning. If you wait another 20 minutes, if we hit this 30-minute line, we've pretty much adapted entirely to being in the dark. This is like you get up in the middle of the night to go to the bathroom, and you can walk to the bathroom in the dark just fine because your rods have completely taken over and you're used to seeing in the dark. So to get to that optimal operation in the dark takes us about 30 minutes. But we have a very quick swap over, and we'll see the most difference in just the first 10 minutes. So we adapt very quickly, but we get better if we wait longer. And for light adaptation, we do the same thing in reverse. If you have to get up in the morning and maybe a sibling flips on the light in your room, it's going to be painful at first if we sort of go backwards, where there's lots of light, but you don't need much light to see, so your eyes hurt. Maybe you're going to squint. Your pupils will be very small because it's way too bright. And so you have to start adapting the other way, where the rods have to get used to having more light than they need, and eventually the cones will kick back in and take over, allowing you to see fine in fully bright light. So the other good example of this is, I've heard it described as movie blindness. If you go to an afternoon movie and come outside and it's, say, 2 o'clock and the sun is shining, your eyes take a few minutes, maybe the whole walk back to the car, to be able to see normally again. And it's going to be a while before your eyes don't hurt from how bright it is. that's your light adaptation, going back to using your cones. Now, as we've said before, color is going to have something to do with the different properties of the waves of light that we're looking at, but we want to make it clear that color is not actually a physical property of the light itself. Color is a psychological interpretation. And what I mean by that is light that is red looks red because that's how our cones and our brain interpret the signal that comes from light that is of that specific wavelength. So that wavelength of light isn't anything special until we interpret it as a particular color. Just a little bit of a technicality, because it's all about our perception of it, our interpretation of it, rather than light flying around being a specific color. So all three of the properties that we've talked about in terms of waves are going to help influence our perception of color. So our wavelength or our frequency is directly related to our hue, what specific color it is that we are perceiving. Amplitude is going to influence the brightness or how much energy is in that particular wave. And purity gives us our saturation. So is it vividly blue or is it kind of grayish, unsaturated blue? So all of these different qualities will influence how we perceive any particular light. And for this section, because we're talking about something visual, you might find it helpful to look at some visual references. So I've got a couple of videos. The first one here is just a 1.5 minute how we perceive light. So giving you a video of what I just talked about. So if that's something that interests you, that's linked up on our eClass page right now. So when we're talking about seeing light and seeing all of these different colors, we're going to be looking at different ways that different colors are interacting. Now, when you think about color mixing and putting different colors together, you usually think about art class. Maybe in grade school, you got the 3 primary colors and you would mix them all together and you get different shades or different colors based on what you mix together. So the idea of red and yellow makes orange is coming from that experience with paint. Now, that isn't the only way that you can look at mixing things. And that's why we're going to talk about 2 different kinds of color mixing. So mixing paints in grade school was a method of subtractive color mixing. So the idea here is that something that is red is going to absorb all of the wavelengths of light except for red. So if you have a red apple sitting on your desk and white light hits that red apple, everything except for the wavelengths that go along with red get absorbed by that object, and red wavelengths get reflected back to our eyes, so we see that object as red. So when we're looking at subtractive color mixing, we're looking at the wavelengths that are getting absorbed by an object. So if you have red paint, then that paint is absorbing everything except for red. So if you mix red and yellow, then it's going to be absorbing sort of everything but red, but also everything except yellow. So you're left with a little bit of red and a little bit of yellow getting reflected to your eye. And so with subtractive color mixing, actually, if I can hop to the next slide, I have an image of it here. So if you have your red and your yellow and your blue, those are your 3 primary colors. If you mix them all together, you end up with kind of a brownish, grayish, blackish mixture. In an ideal situation, if you had pure yellow and pure red and pure blue, you'd get black, because if you absorb everything except yellow and everything except blue and everything except red, you kind of end up absorbing everything. So you subtract all of the possible colors that we could see, and you're left with black. So that method, that color mixing method that we think of, is called subtractive color mixing. And on the next slide, there's another video that goes over the specifics of color mixing, just in case stationary images aren't doing it for you. But subtractive color mixing isn't how our eyes work. So I need you to sort of accept that that's how subtractive color mixing, that's the way that you usually think about mixing colors, but that's not what our eyes are doing. What our eyes are doing is actually additive color mixing. And this is because subtractive color mixing is looking at absorbing everything except the light that gets reflected. Additive color mixing is talking about mixing different forms of light. So putting extra light, so different wavelengths of light together, is going to get us closer and closer to having white light. So if we think of our whole spectrum of light, where it's sort of red on one end, it goes through a bunch of different colors and ends up blue on the other end. If we had multiple wavelengths of light, all of those together are going to start looking closer and closer to white light, because we are adding extra wavelengths. And if we think about white light is the mixture of all of the possible colors of light that we could be seeing. So when we talk about perceiving different colors and looking at different things, we're going to be looking at additive color mixing. So from here on out, we can forget about subtractive color mixing, because we're only really caring about mixing together different wavelengths of light, because our sensory receptors for our eyes, specifically our cones, are going to be picking up on and responding to different wavelengths of light. So that's what we're caring about from here on out. And so this leads us to the idea that our eyes have different kinds of receptors, different kinds of cones that have different sensitivities to light. So far we've just talked about cones generically, where cones are sensitive to light. But now we're going to get into a little bit more detail, and we actually have 3 different types of cones. And these types of cones seem to be differentially sensitive to specific wavelengths. What that means is there is one type of cone that seems to be sensitive to red light. So if we see something that's red, if red light enters our eye, those cones will fire in response to send a signal to our brain that we're seeing something that is red, or something that is the frequency of red. Other cones are only sensitive to green light, so they're going to fire in response to green light, or the frequencies that go along with green. And then our other type of cones seem to be sensitive to blue light, so they're going to fire when they see something that is the frequency for blue. And so by having red, green, and blue, we can have additive color mixing, where even though we only have these 3 different types of receptors, we can still see the whole range of colors that we are capable of seeing. And I actually want to take a brief second and scroll way, way back to this diagram here, because I want to explain why having red, green, and blue gives us a decent coverage of all of the possible wavelengths that we can see. So if you notice, red is over on one far side here. So these are going to be our longer wavelengths. Blue is going to be our shorter wavelengths, and then green falls right in the middle here. And so if we have one type of cone that sees blue, and a second type that sees green, and a third that sees red, then we're going to be able to get a pretty good range of what we're seeing. So if you're seeing light that is specifically this frequency, then maybe you're going to get a little bit of firing, a couple of neural impulses or action potentials from the cones that see blue, but you're also going to get some that are showing green. And our brain can average out, so how much is the blue cone firing and how much is the green cone firing, and it's going to give us this idea that, okay, we're seeing something in between blue and green. It must be this shade. And so from additive color mixing, that's how we're getting sort of the difference between these two. So I always forget every year to move a copy of this slide early or later on so that we can talk about that without having to scroll through. But that's why having red, green, and blue can give us such a complete and complex view of different colors that we are capable of seeing. And so this 3 different receptors, seeing red, green, and blue, are tied to our trichromatic theory of color. And it kind of makes sense here. So tri meaning 3 and chromatic meaning color sensors. Chroma for color. So this theory is tied to those 3 specific types of cones being sensitive to 3 specific things. And this theory helps explain color blindness. So most people who are colorblind are what are called dichromats. So if we're using our roots here, di meaning 2 and chroma meaning color sensors. So in this case, they're actually missing one of the color sensors. So maybe they're missing the red cones and they can't see red light. So they can only see sort of blues and greens, but red doesn't really work for them. And so we can have people who are insensitive to red, or insensitive to green, or insensitive to blue, based on which kind of cones they're missing. So if you've ever seen one of these images, these are a way that you can identify people who are colorblind, because if you're missing, say for this one here, where we have sort of greenish bubbles in the background and orange bubbles in the middle, if you are red insensitive, then the red tones in the number there would disappear, and it would be very difficult for you to tell that there's a number here at all. So each of these different combinations of colors tests for a different kind of sensitivity. Now, these tend not to show up fantastically digitally for whatever reason. Like I find this one and this one kind of difficult to see. So don't panic if you're not seeing the numbers in all of them. Some of them are just really challenging to localize. But sometimes you will spot people who are colorblind who didn't know it by showing them these images. All right, so for a quick example, if someone can't tell the difference between red and green, they might mix up, say, blue and purple, because purple has some red to it. And if they can't tell that it's red, then it's mostly just going to look blue, kind of like this. And I find images like this to be really interesting. So in just a couple of slides, there's a website that actually goes through real life images and shows you what it would look like if you were different kinds of colorblind. So you can go and see what it would be like if you were missing your red cones or your blue cones or your green cones, or what if you were missing more than one type of cone, because that is rare but possible. So a very interesting website to spend some time on, but just for the sake of interest. And on that, for your interest note, I just thought I'd include a couple of extra pointers just because color blindness is such a cool factor. So people who are insensitive to red light, they are determined to have protanomaly. People who are not sensitive to green light have deuteranomaly. And this insensitivity to green is actually the most common kind of color blindness. And people who are not sensitive to blue light are trichromatic. And the insensitivity to blue light is the most rare kind of color blindness. So insensitivity to red is moderately common, green is more common, and blue is less common. And again, this is just for your information because I think color blindness is kind of cool. So I did a little bit of extra digging. And this is the website that I was talking about. This is also linked on eClass if you wanted to spend some time playing around with images that show different kinds of color blindness. So that's one theory. Now, I know I kind of talked about it as if that was the only theory, but in fact, we have another possibility, a competing theory, as it were. And so this is going to be the opponent process theory. And it's going to start off very similar to a trichromatic theory, but then it's going to change very, very dramatically as we get towards the end of the explanation. So opponent process theory, just like our trichromatic theory, starts with the idea that the human eye has 3 different types of color receptors, 3 different types of cones, and they have differing sensitivities to different wavelengths. Nothing's too dramatic there. And the reason why they're both so similar at the beginning is because we have 3 types of cones, and those types of cones are structurally different. So if you look at cone cells under a microscope, you can tell them apart. But now they start to differ because the opponent process theory actually says that each cone is responsive to 2 different wavelengths. So unlike our trichromatic theory, where we say that one cone responds to one wavelength, our opponent process theory says that each cone is going to act kind of like a switch, and it's going to respond to one of 2 things. So our first type of cone is going to send a signal that says red, or it's going to send a signal that says green. So these cones, our type 1 cones, will tell our brain if it's seeing red or if it's seeing green. Our type 2 cones, the second kind we have, will send a signal to the brain if they're seeing blue or if they're seeing yellow. And then our third type is going to send a signal if they're seeing black or white. And this is more of a, is it bright or is it dim? So this one would be giving us more of an idea of the intensity or the brightness of something rather than black or white, just based on white being the presence of many different wavelengths and black being the absence of wavelengths. So lots of energy or a little bit of energy. And so what we're saying here is that any one cone can act as a switch, and it's going to say this one or this one. And so you might be thinking, well, if our trichromatic theory explains color blindness and it seems to work, why do we have another theory? Why does this one even exist? And the reason is, opponent process theory helps us explain a weird phenomenon called after images. So if you went to school in Canada, you have seen this image before, where they get you to stare at this black dot in the middle. You hold your head very still, stare at that black dot for 30 seconds, and then you look at a white piece of paper. And if you stare long enough and then adjust your perception, you would adjust where you're looking. When you look at that white paper, what's black on the screen looks white, and what's green on the screen looks red. If we switch back here, what was green now looks red, and what was black now looks white. So this is kind of explaining that switch showing us one or the other. So when we have an after image, they're going to be these persistent ghost images that stick around after we look away from a stimulus or after that stimulus is removed. And the idea here is that if you look at something that is green for a really long time, and then you look away, this cone was saying green to the brain for so long, it's sitting there saying green, 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 that it gets tired. And when that green goes away, it overcompensates. And instead of saying not green, it goes all the way to red. So it kind of jumps the other way and sends a signal to the brain that says red. Or if it was saying that something was black, it's going to overcompensate after a while and say white. So the opponent process theory helps us explain after images, which all of us experience and is a pretty big thing that needs to be explained by the theory that works with our vision. So now we've kind of hit a problem. Our trichromatic theory explains color blindness, and our opponent process theory explains after images. So which one is correct? Which one is closer to what we actually have? And the answer is a bit of both and also neither. So we come over to our dual process theory, and this is as close to what we think is going on as we can cover in this course. And this dual process theory is going to combine both the trichromatic and our opponent process theories. So dual meaning both of them. So we still say that yes, we have 3 different types of cones. They are still each sensitive to different wavelengths because, like we said, we have 3 different structural types of cones. But in this case, that opponent process side of things, that blue or yellow, red or green, black or white, doesn't come from the cones themselves, but from some of those support cells that we kind of glossed over when talking about the structures within the eye. So our lateral geniculate nucleuses, or LGN cells, are basically going to help filter or respond to what our cones are seeing. And this is one of those things where an image is going to make it a lot clearer. So for our dual process theory, we have our 3 types of cones that are conveniently cone-shaped. So ones that respond to blue, to green, and to red. But before those signals from our cones, from those receptors, get sent to the brain, they get processed just a little bit within the eye. So these are our LGN cells, and they're going to be getting signals from these cones. So there are blue-yellow opponent LGN cells that are going to tell the brain blue or yellow. And so blue is just getting a signal from blue, but yellow is going to be a signal from both green and red. For our green-red opponent LGN cells, they're going to send a signal to the brain, but they're going to tell them either green or red, based on what incoming signal they have. And the same for the black and the white. And so I'm sure you can imagine that this process can be very complicated if we wanted to delve into the specifics in how they are filtering this information, but we're just going to keep it very high level for the purposes of this course, because again, we can't really afford too much time on each specific one. But this is a very simplified version of what we think is actually going on within our eyes. So the dual process theory is closest to accurate in terms of those 3 theories. Now, this has been really biology heavy so far, talking about the structures of the eye and how we're perceiving things. So let's dabble just a little bit in some more psychologically minded stuff, at least for a little bit. So if we wanted to look at influences of color, so how does our perception of color influence our behavior? That's a pretty good way of applying this to psychology. Now, most of our examples are going to be talking about the color red. And that is because red seems to be something that is very biologically relevant. We seem to be hardwired for a lot of things to do with red. And so red seems to be one of the easiest colors to study when it comes to psychological effects. At least I haven't found a lot of good research that looks at other colors. So we're going to be talking about red and how red influences our behavior. So in this paper here, they talk about the fact that the color red can undermine performance in achievement contexts, which is a kind of fancy way of saying that if two sports teams are playing each other, the team that wears red ends up having an advantage, not because the team wearing red performs better, but because the team that isn't wearing red perceives the team in red as more of a threat. So the people not wearing red don't try as hard because red is intimidating and it makes them think that they don't have a chance to win. And that's a very simplified version of this research, but that's sort of the basics of what they're getting at. Another study looks at how red affects our performance. And red can actually reduce our performance because it disrupts our attention. So if you think of things that are red, kind of catch your eye. It's why our fire trucks are painted red. Ambulance lights, one of them flashes red. It's meant to draw our attention. So they can actually quantify how distracting having something that's red can be. And red also seems to be related to attractiveness. So people wearing red tend to be rated as more attractive than people not wearing red. And those are some really interesting studies. And even red is kind of like we said before, it's attention grabbing, but it's also easier to hold your attention. So a red target on a computer screen is easier to follow with your eyes than any other color. So red seems to be something that we are pretty much hardwired to pay attention to, which is kind of cool. So back to the biology for a little while, but this is building us up to something really cool to talk about. But the same way that we had talked about our ability to localize sources of sound, our eyes have the ability to perceive depth. So we have what's called depth perception, which lets us interpret visual cues to tell us if something is close or far away. And we have two different types of depth perception cues. So we can have monocular or binocular cues. And we can use our root word investigations here, mono meaning one, ocular probably meaning eye. So this one is something that could be done with only one eye. And binocular means two, and eyes still is ocular. So in this case, you'd probably need both eyes to perceive that effect, which is exactly the case. So we're going to start by talking about binocular depth cues. And so these are based on the fact that the image coming into our eyes differs between both eyes. So you can see this yourself by sitting exactly where you are. If you close one eye and leave the other open, and then switch so that the eye that was closed is now open and the eye that was open is now closed. If you switch which eye is open and which is closed, your world seems to shift just a little bit. And that's showing you that one eye sees the world from a slightly different angle than the other. So this property is called retinal disparity. Objects are projecting images to slightly different places on our right and left retina. And this happens because our eyes are spaced just a little bit apart on our faces. So our right eye and our left eye are seeing the world just a little bit differently. And our brains can interpret those differences, where if something looks very, very different between our left eye and our right eye, it's probably close to us, so that we're looking at it at a more extreme angle. Whereas if something is really far away, then it doesn't really matter if you're looking at it with your right or your left eye, because it's very far away. We also have something called convergence. And this is the idea that if you're looking at something that's close to you, your eyes are going to curve inward just a little bit to be able to see that. And I'm actually going to doodle on here so that we can see that. So I'll be a good artist and tell you what I'm drawing. We have our two eyes, our left and our right. And if we're looking at an object that is fairly close to us, our right eye is looking directly at it, and our left eye looks directly at it. And there's a little bit of an inward angle, that convergence, as the eyes have to focus inward to see the object. Whereas if we had, get rid of that, if we have an object that's further away, then that's a really bad line. Our convergence is much less. The angles are less steep, or they would be if I was a better artist. So the further away something is, the less convergence we experience, and also the less retinal disparity. And our brain can interpret those two factors to help us decide how near or far away something is. Our next type of cue are those monocular depth cues. So these aren't cues that you can only see with one eye. It's just implying that you don't need the use of both eyes to perceive them. So this isn't based on the fact that we have two eyes. It's just based on how we perceive things in our world. So the first of these is motion parallax. And this is looking at how objects move across the retina. And the best way to envision this is think about sitting in a car. You're at the window of a car and you're watching things outside the window fly by as you're driving. So the things that are very close to the car, maybe the grass on the side of the road, is really blurry and fuzzy because it's moving past you very, very quickly. But trees that are off in the distance don't seem to be moving very quickly at all because they're further away. So that difference in perceived motion is motion parallax. So you can learn that things that seem like they're moving faster are probably closer to you, and things that seem to be moving slower are further away. And we can also use what are called pictorial depth cues. And these are a really good way for giving us depth perception without having to use both eyes. And if you've ever taken an art course, basically everything they've taught you about perspective and drawing things with depth, they all fall under these pictorial depth cues. So things like linear perspective. You've ever seen those pictures where you draw a horizon and there's train tracks that vanish into the distance. That is linear perspective, showing that it gets smaller as it goes further away. You'll also see texture gradients, where things that are closer to you have more detail because they're closer and you can see them better. There's interposition. Things that are nearby are going to cover up things that are further away because they are closer to you. We can also look at relative size. A person standing near you is going to look larger than someone standing farther away. And even things like light and shadow, intensity of color, all of those things are all pictorial depth cues, and they give you a sense of how far away something is.